I like to start all my talks with this slide because this is a Arcambolo face, and Arcambolo was a 14th century Dutch painter who made faces out of fruit, still life faces. And I had found a contemporary guy doing faces out of whales. He was a marine biology student named Chris Oxley, and I asked him if he would do an Arcambolo face with mushrooms for me. He said he didn't know anything about mushrooms, so I designed a sort of nightmarish face of mushrooms modeled on myself, and it underlies how obsessed with mushrooms I am and what makes me fascinated by fungi. I have what's kind of turned into this crazy social media empire of like almost 3 million cumulative followers across all these different platforms, which is an overwhelming number for the most part. And the reason I got to this point is not because I've stuck with doing straight educational content. It's because I've done entertainment with mushrooms. And just kind of give you guys a little flavor of what that looks like, I'll warn you, this is what I would call emotionally evocative educational content. So this is a St. Patrick's Day compilation I did a couple of years ago, and it's just sort of like quick, mushrooms in your face kind of thing. So ooh, I got a little music tapping on a mushroom. There's some plasmodium pulsing. We have a, Hydnellum peckii. There's some chanterelles. There's a slime mold. There's a jelly fungi. There's Piziza cups. There's some lichen. There's more slime mold pulsing. There's a slug eating a mushroom, banana slug. There's some parrot mushrooms. There's a Hydnellum peckii getting squeezed. There's some Chlorosiboria, microheterotroph, droplets, ink caps, and puffballs. And so that video has been viewed like 40 million times. And it's kind of a wild thing to try to figure out, like, why has one piece of content been viewed so many times by people worldwide? And I think the answer is because it's kind of quick and in your face, and people go, what is that? And the idea of what I'm trying to do with my content is to cast a wide net and funnel people in to the more educational stuff that I've done, which is, more informative videos, as well as the captions that I write on most of my posts, where I'm giving more context for the mushrooms that people are seeing and talking about edibility and identification and that kind of thing. But you got to catch their attention first, because that is the currency on the internet. Is you have about 3 seconds to get someone to actually watch your video. And so that's part of the idea of what this is. And I started originally sort of on Instagram, and it spun out into all these different platforms and grown more than I ever could have imagined. So my gateway mushrooms and the things that got me into mushroom hunting in the first place started in childhood. And I had sort of a long gap there between, when I was a kid and when I was adult and I got into mushrooms. But it started early for me, and it was in the woods of northern Quebec at Lake Tremblant. My grandparents had a cabin, and I remember walking through sort of the soft forest stuff and coming across these slimy little green mushrooms. And I'd seen mushrooms before. I was aware of what they were, but finding little slimy green ones was sort of a life changing event for me. And it stuck in my mind, where I was like, what are these things? And I was dead convinced they were an alien. We took them back to my grandfather, and he was like, I think those are, parrot mushrooms. They're a kind of wax cap. And I was like, BS, they're an alien. But I was four. So, the other thing that I remember, particularly from being a kid, was my mom and I were out for a walk, and we found a nice big puffball. And she knew enough that said, "Oh, we can definitely eat this if it's nice and all white inside." So we took it home, sliced it up, and threw it in the pan with a little bit of butter. And the first taste of that kind of like marshmallowy, soft tofu texture and really nice savory flavor, like absolutely like struck me. And I remembered eating it and was like, "Wow, that's so good." And I've been chasing the dragon ever since there, trying to find another puffball that was as good as that one. Uh, Another experience from being a kid was we had a big stump in the yard, and my dad had decided he didn't really want to get it ground down, so he looked into buying some plug spawn, and he ordered chicken of the woods plug spawn. We drilled some holes, tapped them in. I was really excited, but I was like 9 and immediately forgot about it because I had the attention of a goldfish, or memory of a goldfish. And it was about 2 years later, I think, that this brilliant yellow and orange polypore mushroom came out. And polypores are, not a cap and stem mushroom. Instead, they kind of are often growing out in shelves, and they have lots of little pores underneath. So chicken of the woods is very distinctive. And I was, clearly excited about this big orange mushroom. And I asked my dad, can we eat this? And he's like, well, he's very safe. He's a scientist, but he's like, we inoculated this. It looks like the picture in the book. So yeah, I think we can eat this. And I remember cooking it up and being really excited, but I didn't quite know what to expect. And being called chicken in the woods, I had an expectation for it to taste like chicken. And in reality, chicken in the woods doesn't really taste like chicken. It has a slightly like woody, slightly sour polypore kind of flavor, but it really does have chicken-like texture. So when you fry it up in a little bit of butter and salt, it does taste a lot like chicken because texturally it feels like the muscle fibers of chicken in your mouth. And that was a really compelling moment for me that made me really love eating mushrooms, particularly like these sort of wild mushrooms. So I was all set up to go on a foray with the Boston Mycological Club that fall, and disaster struck because I became a anxious pre-teenager, and everything that I thought was cool previous to this point suddenly became, profoundly uncool, and I was like no longer interested in going to the woods or doing mushroom stuff. So I forgot about mushrooms for about 20 years, and it wasn't until I had finished my PhD and I was living in New Zealand and I started going for hikes and I was seeing stuff all over the place and getting kind of fascinated by all the nature I was seeing because it was so different from where I'd been in Massachusetts or in California. And I came across this kind of fuzzy brown mushroom. And if you guys are familiar with a piapini, that's basically the New Zealand version of a piapini. But I saw it, and when I first found it, the partial veil was on, so I didn't know enough to know that it was a partial veil. I just thought this mushroom didn't have gills. So I'm Googling, brown mushroom on a tree without gills in New Zealand, and it wasn't having a whole lot of luck. Because I think if you've ever tried to search for a mushroom by a sort of a pure description of what it looks like, you're not going to have a lot of luck unless you're trying to find Amanita muscaria, right? If you search for red mushroom with white dots, you'll find muscaria. Almost anything else, you're probably going to end up with the wrong mushroom from the type of description you would give, because as a beginner, you don't know the specific mycological words that help you kind of key in morphology and figuring out what something is. So what I did was I uploaded this photo to my Instagram, and I just asked my friends, "Hey, what's this?" And it was a friend of a friend from college who'd sort of seen it and then said, "Hey," suggested to me, compare it with this thing that I had seen in New Zealand, and it was called, the local name was Tawaka, or Cyclocide parasitica. And like I said, it was a piapini mushroom, and I was diving into reading about this thing and realized I was like, okay, well, in New Zealand, there's nothing else that looks like this. There's no toxic lookalikes. As long as I know that it's sort of brown fuzzy mushroom that's growing out of a tree, I think I'm pretty safe to actually try eating this thing. So the next time I found it, this was, somewhere out by Lake Taupo in New Zealand, and I'd moved to Hawke's Bay. And there I started foraging a bunch because I was finding these mushrooms and I got really excited about picking them, bring them home. I was really trepidatious at first, but I cooked them really well, tried a small piece, waited 24 hours, tried some more, and realized how delicious they were. Next thing I knew, I was putting them in all of the food I was making. And particularly, I really liked getting fish and chips from like a local place and then making tacos with the fish and putting mushrooms in it. And it was a fantastic dinner. So that got me really turned into being excited about foraging and, excited about the free food I could find in the woods. And I was, floored by like, wow, how many mushrooms are there out there that I haven't tried? And that was this like big black box for me that I was so excited to kind of dive into as a beginning forager. So this was my gateway mushroom for foraging. And then the other thing I saw in New Zealand all over the place was called the orange pork conch, or Favelocia calicera. So this is a little orange mushroom that was all over the dead and decaying wood in New Zealand, and I thought it was really pretty. And if you scroll all the way back in my 2000 plus Instagram posts, this was like the very first mushroom I posted was this crazy cool orange one, and it got like 250 likes, and I was like, wow, there's really something to this posting mushrooms online. And I started a sort of a mushroom only account called, Fascinated by Fungi, and that's where I was doing it. But I was reading about the mushroom and I realized that it was invasive. Was actually from South Africa. And so this taught me something because I was like, wow, this thing looks so cool and I was so fascinated by it, but it's actually invasive and it's clearly out of balance in this ecosystem because it's growing so well that it's forcing out and depleting the biodiversity of the stuff that should be here. And so at the beginning of my mushroom hunting career, I was really into both the stuff I could forage and eat, as well as trying to understand kind of the ecological relationships and the science of what I was finding in the woods. And that's part of why I became so fascinated, was what I had in childhood and then the things I ran into as an adult. So who am I and where do I come from? I'm from Cambridge, Massachusetts, and my parents live on the North Shore, north of Boston by about an hour. If you've ever heard of the Perfect Storm, they live right beside Gloucester, Massachusetts, in a tiny town called Rockport. And I go there in the summer and mushroom hunt. It's wonderful. But around 18, I went out to UC Santa Cruz and got a biochemistry degree. While at Santa Cruz, I did some undergraduate research on malaria through a Howard Hughes lab. I also got really into scuba and sailing. And then, as a function of sailing, our sailing team captain was a brewer, and we had way better beer at the sailing team parties than there were at any other college party. So I was like very interested in learning about brewing because I was like, I like good beer. And out of my interest in brewing came an intense fascination with yeast, the single-celled fungi that does fermentation. And I was really fascinated by how can I use different strains of yeast and different fermentation conditions to influence the style and flavor of the beer that I'm making. So with that in mind, I applied to UC Davis, where I was really excited about like the food stuff, the science stuff, the fermentation kind of like things they were doing. And I found a professor, PI advisor named Linda Basson, who's a really epic and awesome yeast geneticist, and, as well as being an incredible wine educator. So I learned a ton about wine fermentations from a technical perspective, but also how to taste and talk about wine, which was a whole sort of secondary education that I wasn't counting on. So after 7 years at Davis, I finished my PhD, and I was like, you know what? I say that I have this sort of biochemistry background, and I've worked in wine, but I haven't actually done a real wine harvest at a big commercial winery. So I went and got an internship at Opus One Winery, which is kind of like a celebrity branded winery. It was a collaboration between the Rothschild and Mondavi families that came together and started this brand called Opus One, where they make a Bordeaux style chateau or chateau style wine. And out of like 80 different lots, they blend them all into one wine that's very special and sells for like 400 plus a bottle. This is high end cold wine in the Napa Valley. So they had money to invest in some research and having kind of interns doing special projects. So previous to me showing up there, they'd been fermenting most of their wines with just one yeast. And while I was there, I helped to isolate a bunch of yeast that corresponded to different vineyard blocks. So as they were bringing fruit in, I would prepare and grow up yeast to ferment the grapes that were coming from a particular block with the yeast that came from that block. And the whole idea was adding sort of layers of terroir and complexity to the overall wine and blend and adding more kind of aromatic nuance to the wine. I was also pioneering some new fermentation tech and probes that we were putting in the tanks to kind of measure what was happening with the yeast and fermentation. Out of that technology work, I started working in Bay Area biotech sales, and I was going into lots of cool pharmaceutical companies and food processing places and kind of next gen biotech. I was in at Impossible Foods, where they make those kind of cool burgers. They would use plant proteins to make them kind of bloody. So I was learning about all this stuff. But I was getting a little burnt out by doing sales because I was having to like send emails or like, hey, I'm just touching base and checking in. I want to like, see your thoughts on those quotes. And it was starting to kind of like kill me inside because I'm like, I'm getting better at sales, but not getting better at science. So I ended up going back to UC Davis and I did like a 50% postdoc where I was looking at some of the underlying technology and trying to understand what was the chemistry of terroir. So we were using some of that stuff I'd been doing at Opus One and what I was doing in my sales job to really look at wine in this very mechanistic, scientific kind of way. And then COVID happened and everything kind of petered out. I managed to publish a paper through the postdoc, but it kind of didn't evolve the way I was hoping it to. It didn't lead to an academic job. And the sales thing, I was like, I don't really want to go back to sales. So I decided, hey, I'm going to leverage the following I have on social media and write a book proposal, get an agent, and pitch it. And I got a book deal that was pretty substantial, allowed me to go full time on being a mushroom educator and leaning into doing the social media while I was writing a book. So that's why I'm here now. And this is what I'm doing. Beyond mushroom hunting, I really like to cook. I forage for other stuff other than just mushrooms. I garden. I love the ocean, and I really like my cat. There's Honey Badger down there smelling a couple shiitakes. He's a little bit of a menace, but he's a good dude. Okay, so I wrote a book. It's called Passport to Kingdom Fungi. And the big question I get from a lot of people is, well, how is this different from any other mushroom book, right? There's a lot of great mushroom books. How is yours different? And the thing I wanted to do was a lot of mushroom books are great, but if you open them up and you have a mushroom in front of you, all you can really do is kind of flip through the book and look at the pictures up until you find a picture that matches the mushroom. And if you don't find that and you get to the end of the book and you're like, well, I didn't find a mushroom that looked like the thing in my hand, I don't really know what to do. And so I wanted a book that had a little bit more structure in terms of teaching people about how to recognize and bin the finds. So you weren't just sort of sitting there endlessly flipping through looking for something that matched. And I wanted to make it really accessible too, right? I wanted a book that people would pick up and look at a lot, not just something you read through once and then it sits on a shelf the rest of your life. So I packed it full of photos and really cool infographics and science illustrations, which you'll see some of those during this talk. And I wanted it to cover not just mushrooms, but all kinds of fungi. So I'm sure you guys know this because you're in my ecology club, but all mushrooms are fungi, but not all fungi are mushrooms. And so there's maybe roughly 150,000 species of mushrooms, but there's probably 2 to 5 million species of fungi. And we only know about like 5 to 10% of those have been sort of sequenced and described, but there's this huge unknown of like what's out there in terms of fungal dark matter. And I wanted to kind of get into some of that so you guys have a better idea of like how broad and interesting the fungi kingdom is beyond just mushrooms. And trust me, I talk about mushrooms, but I'm talking about some of the other stuff too, because they are interesting. So I dove into the biology, the ecology, the biochemistry of fungi, as well as identification and foraging and cooking and all that kind of stuff. And I see in my ecology there's a little bit of an issue with hero worship. Certain people get sort of overrepresented in the world of mycology in a way that I don't necessarily think is fair. So one of the things I wanted to do was take the focus off myself and bring other people into the project. And so throughout the book, I have little blurbs from different important mycologists who are friends, and I think they're really what they're doing for the field of mycology is really important. So I got their perspectives added to the book, and I really focused on getting diversity of perspectives too. So it wasn't just people who look like me. There was a lot of different thoughts and ideas and backgrounds that went into the people I was reaching out to. So the format of the book is basically the first half is question answer. There's 10 chapters of what is a mushroom, what is mycelium. You kind of ask a question, you get like a paragraph sized answer. So you can jump around if you want, or you can read it cover to cover. Either way, whatever works for you. And the second half of the book is focused on kind of fungal profiles, where I give information on foraging, identification, and in a lot of cases, culinary notes as well. And then every single listing in the book has a QR code, because you don't have to be dependent on the 3 or 4 photos I was able to put in the book. You can hit the QR code and go look at thousands of photos on the internet, specifically on iNaturalist. And I covered things at the genus level in kind of a general sense, because whether you're in Minnesota or you're in Massachusetts or you're in Asia or Australia, if you find a chanterelle, a true Cantharellus, you're going to be able to eat it. And so I'm less concerned about species identification and more concerned about broad categories of mushrooms. And that's how I organized the sections, and that's where the QR codes go to as well. So you can look at thousands upon thousands of photos of Cantharellus and recognize what a true chanterelle looks like pretty quickly by just comparing the photos. So that's all the stuff I did, as well as including non-mushroom fungi and some of these different categories. So we'll talk about them as we go along here. Okay, so here the book starts out with, where are fungi and what do they do? There's some really fundamental questions that I find a lot of people don't necessarily think about as the first thing, but I think it's important to kind of set the stage for everything else we talk about. So as a mycology club, you guys are probably pretty familiar with the idea of saprotrophic mushrooms, ones that will decay things and break down organic matter to recycle nutrients back into the soil. And you're probably familiar with mycorrhizal mushrooms that live in conjunction with trees and colonize the roots and help them access more water and nutrients in the soil. What I didn't know about and hadn't fully thought about was all the other places that fungi show up and all the other forms they show up in. So there's fungi that are living on the leaves of plants as epiphytes. There's fungi that are living inside of plants as what are called endophytes, kind of like the bacteria in our guts. There's fungi that are in the air. It's constantly spores in the air that you're breathing in and out. And for the most part, they're fine. They don't affect you. There's, fungi in the stratosphere going, in all the air currents from continent to continent. Every single aquatic and water system on the planet has fungi in it. And they're not there at super high amounts compared to some of the other organisms, but they are there and their influences are profound. So there's fungi in the ocean, in ponds, in salt marshes, in the deep ocean at like hydrothermal vents. Pretty much anywhere and everywhere you look for life, you're going to find fungi, which is really amazing because we don't normally think of fungi like that. We have their spot in the forest and the soil, but we don't think about them participating in the ecology of stuff like deep sea vents, but they are there. Animals have fungi associated with them. So there's a whole class of fungi that are associated with digestion in ruminants and even birds and reptiles as well. And those are things that like obligate live in the gut of those animals and help them digest plant matter. We as humans have fungi all over us. We have fungi in our skin, and we have, I tell you, everyone listening to this presentation has about 10 million yeast living on your scalp right now. And I know that sounds a little weird, but it's a good thing because those yeast are there eating some of the oils that come off of our skin, and they're there displacing other bad stuff that could fall onto our skin, and they'll sort of muscle them out because they're like, hey, this is our habitat. This is where we live. However, if you get an overgrowth of that yeast, that's what dandruff is. Even an overgrowth of that yeast in your skin, you can get depigmentation, lightening of the skin, that thing that happened to Michael Jackson. So those are examples of places that you may not have thought of fungi existing, especially in conjunction with animals. Speaking of animals, there's the idea of what are called entomopathogenic fungi. These are fungi that target insects in particular. One that you guys have probably heard of is cordyceps. So it's a particular type of entomopathogen that can infect, in this case, let's say ants, and it grows mycelium through the ant's body and then causes them to like march up a branch, latch on, and then grow a mushroom out of its head and rain spores down to like continue the life cycle, which is a pretty wild thing. And it's the basis for the sci-fi show and video game called The Last of Us, where cordyceps jumps the species barrier. And to spoil the whole thing, it's not really possible that it's going to jump to humans, but it is really cool to watch it happen in insects and understand all the different kinds of entomopathogenic fungi. And then there's lichens, which are a symbiosis there of fungi and then like an algae, and the fungi is providing the structure and the algae is doing the photosynthesis. And then there's a whole bunch of other stuff involved in that symbiosis that kind of makes it like a whole holoorganism, like a little forest unto itself. And there's even fungi who live inside of rocks. That are mineral munchers, and they can live for like 10,000 plus years. They grow very, very slowly, but they are slowly breaking down minerals and rocks. And these things are like in Antarctica, living inside of like very extreme conditions, but they are out there. So fungi are at every single interface in life, and they're interacting with everything. And that's because they've evolved along with everything else. They've evolved in conjunction with all different forms of life. And the role that they play in our world is really fundamental because there are primary producers, which are plants. They're doing photosynthesis, producing sugars from the energy of the sun. And then it's fungi and bacteria that are breaking down the plants to create life. And fungi, I like to think of them as they set the table for everyone else. So bacteria really like to eat nitrogen, and fungi are really good at decomposing complex carbon. And between the action of those two organisms, they're able to turn most things back into stuff that something else can eat. And then that allows trophic levels to kind of build from there. So, one thing eats another thing, eats another thing, eats another thing, and we get all the way up to like, tunas and sharks and humans, which are sort of apex predators. But there's a whole lot of life that happens there. And all of it is facilitated by the action of fungi helping to break things down and make things bioavailable again after plants have produced it. So it is not an understatement to say that fungi are keystone organisms on our planet, and we would not have a planet the way we think of it now if it were not for fungi. So they're incredibly important. And it always kind of like raises the hair on the back of my neck when I hear the phrase, mushrooms are going to save the world. Because I'm like, well, we already live in a fungal world, right? We would not be here and we wouldn't exist the way we do if it were not for fungi. So I think it's really important. And what I wanted to do with this book was to kind of bring fungi to the forefront and get people thinking about their profound roles in our world and understand them better, to better empathize with what they actually do. Because mushrooms are great, but understanding fungi as a whole brings so much more value to a bigger picture. Okay, so what do the forms of fungi take? Let's start by talking about some of these differences that are fundamental between plants, fungi, and animals. So plants are autotrophic in that they make energy from the sun, do photosynthesis. They use cellulose as their structural carbohydrate, and they use amylose as their way of storing sugar. Amylose is starch. So you're pretty familiar with like potatoes and rice, corn, stuff like that. Fungi and animals are more similar to each other than they are to plants, but there's still about 1 billion years worth of evolution between fungi and animals. So while they are more similar to each other than they are to plants, there's still quite a few differences between them. The things they do have in common is that animals and fungi will use the polysaccharide chitin, which is not in humans, but it's in a lot of arthropods and shellfish and stuff like that. That's what they use to make the exoskeleton. And then animals use glycogen as a way of storing energy. And that's what we have like in our liver. So if you're going for a run, your body needs energy. It'll mobilize the glycogen to give you glucose for your run. The big difference there between fungi versus animals is fundamentally how they eat and digest. So fungi are called osmotrophic in that they excrete enzymes into the world around them. They live in their food and they digest the food, and then they absorb the nutrients internally. Animals are called phagotrophic, and then they have to engulf and eat something and then digest it internally. And there's a lot of things that kind of like blur the line between what's animal versus fungi versus sort of protists and amoeba type things. And the real difference there is what has chitin in the cell wall, as well as what is osmotrophic. That's what defines sort of true fungi. So in terms of the forms that fungi take, and this is something I didn't fully understand until I really started working on the book, was that um, this is a little bit of a brute force scheme, but you have aquatic parasitoids, which is sort of the original evolutionary form of fungi that came from water, and then they evolved to land, and they have couple of different forms on land. So There's yeast, which are single-celled fungi, but they do not form true filaments or networks. You have molds, which form networks and filaments, but do not form mushrooms. Then you have mushrooms, which are macrofungi. The other types are microfungi. So mushrooms are macrofungi. You can see them, and they are filamentous. And then you have lichens, like I talked about, which is that symbiosis. So they're filamentous, and they are living in symbiosis with some sort of photosynthetic organism. And the thing I didn't understand was that the same fungi can have different forms. So there's things out there that are both a mushroom and a yeast, or a mold in a mushroom, or like a lichen in a mushroom. There's things that are lichens and yeast at the same time, and they're different when they're at different points in their reproductive cycle and their life cycle. So an example of this, and you guys are probably all familiar with something like witch's butter, the little kind of orange jelly fungi you see. So in its sexual reproductive, what's called a teleomorphic form, that's the mushroom form. Witch's butter is that little bit of orange jelly. But in its asexual clonal form, what's called the anamorph form, it's actually a single-celled yeast. And that single-celled yeast can actually be like a human pathogen. It's called but a dimorphic yeast, the thing that can infect humans as a disease. And I, as a human, have jiggled probably more jellies than almost anyone alive at this point. And I've never had a health issue with touching jelly. So don't worry about touching a witch's butter. But it is kind of cool that in these different phases of its life cycle, it can be both a mushroom and a yeast. And I think that's really neat. So I was then asking, like, what are mushrooms made out of, right? So mushrooms are composed of mycelium, this kind of hair-like threads that are often in the soil. And when they come together to form a mushroom, they felt together into much more dense structures. But that is, a mushroom is still fundamentally composed of mycelium. And mycelium is composed of fungal cells known as hyphae. That's the individual unit of a fungal cell. If we zoom in on the edge of a hyphae, on the cell wall and the cell membrane, we see these two structures. So the cell wall is like a scaffolding of polysaccharides and proteins on the outside of the hyphae that gives it tremendous amount of strength and adaptability and ability to kind of like infiltrate into rocks and things like that. And then the cell wall is a fluid phospholipid bilayer that's packed with all sorts of proteins that allow yeast to interact with and sense their environment, as well as import nutrients and export waste. So that is kind of the fundamental structures of fungi. And this is a traditional scheme kind of compared with a far more complex way of looking at fungi. So historically, there was like four phyla of fungi. Now there's upwards of 20, but this historical classification is still really useful. So I put in these opistonauts. These are these sort of phagotrophic sister lineage of fungi. These are things that do swallow their food and can have like cellulose in their cell membrane. So they're not true fungi. They're sort of related. But for the purposes of this book, I focused on the true fungi, the ones that are osmotrophic. So the original kind of fungi, like I said, were in water. They're those aquatic parasitoids known as Chytridiomycota, and these things are still around. They have little swimmy spores. Then they lost their flagella and came to land and became filamentous, so they usually produce like one big spore. And then we have the two phyla that I think are sort of the most interesting and the most compelling because they produce mushrooms. So this is subkingdom Dicaria, which contains Ascomycota and Basidiomycota. So ascos and basidios is what I'm going to call them for short. Ascos have a kind of like balloon-shaped structure called an ascus, and that's where the spores are. And this is an example of like a little cup mushroom. If you were to blow into the cup mushroom, it's lined on the inside with lots of those asci, and they're pressure sensitive. So if you blow into cup mushroom, you'll see like 2, 3 seconds later, it'll poof out a whole bunch of spores because all of those asci are loading up minerals and then bursting forth the spores to shoot them out into the air. Basidios, which make up the majority of the kind of fleshy mushrooms that you're most likely familiar with, instead have a club-like structure, the spores in the end there, and those are dispersed through a mechanism called Buehler's drop, which is sort of like a catapult, basically like a water drop catapult that flings the spores off into the air, and they're able to disperse. So the big differences there between ascos and basidios is just their spore types. And this is, like I said, this is a fairly simple scheme, the ABCZ scheme of the ascos, basidios, chytrids, and zygos. And then the beautiful illustration here, the fungal tree of life, was done by Laszlo Nagy, one of his students, an artist named Beta Bus. And it starts all the way down at some of the basal stuff of like little swimming chytrids. There's an example of a fruit fly being eaten by an entomopathogenic fungi. On the left side here, it goes up into the more complex basidios. So you see stuff like chanterelles and witch's butter and stinkhorns and romaria. The coral mushrooms, you see Amanita muscaria, the red and white mushroom with the little dots. You see stuff like porcini. So this is a basidios make up. I think there's about 50,000 basidios, and about half of them form mushrooms. Whereas for the ascos, which is the side here on the right, this represents some of these sort of less recognizable types of fungi. And there's maybe about 93,000 uh, species of ascos, and only a small proportion of those are going to form things that we would recognize necessarily as mushrooms. And there's a lot of forms that are sort of in between, like what's a mushroom, what's a mold, what's a lichen. It's not entirely clear always. But I figure if we're going to call morel mushrooms, we might as well call a lot of the other fruiting body structures we see in Ascomycota mushrooms as well. So that was the kind of logic I went with in this book. But that being said, there's a lot of weird forms in Ascomycotis. You have cup fungi, you have little discs, you have stuff like cordyceps and little club mushrooms. You have a lot of lichens or ascos, and even the yeast that I worked on for my PhD, Saccharomyces cerevisiae, is an asco. So this is an amazing diagram I recommend you guys check out as the fungal tree of life. You can even order it as a poster. It's very cool. Okay, so this is a breakdown of fungal ecology. And all of this is symbiosis. So there's the history of science is often dictated by a bunch of old white guys, and they had said traditionally in biology that everything was in conflict. And it took a woman to come along and suggest that, hey, maybe sometimes things aren't always in conflict, and maybe sometimes they get along. And she was studying lichens, and very much to her point, as they unfolded the science around lichens, they realized that these things are actually living in a mutualistic situationship instead of it being kind of like always at odds with each other. So whether it's good or bad, this is all symbiosis. And there's things that are mutualistic, where both parties are benefiting. Examples of that would be like mycorrhizal fungi, like we talked about the fungi living on the roots of trees and plants, helping them absorb nutrients. The plant is feeding the fungi. You have lichenized fungi. We talked about this, the symbiosis of those things. And animal digestive fungi, ones that are living inside of animals and providing benefit as they're digesting and eating the food inside the animal. But the original way of doing business for fungi was to be pathogenic, was to be parasites. And over long periods of time, it doesn't behoove you to be parasitic on your host. You also, because you're going to ruin where you live. And so a lot of things have over many eons evolved to be less pathogenic and sometimes even mutualistic. So a lot of these mutualistic relationships arose out of parasitic relationships that just, it was kind of an arms race to the point that they realized, well, maybe we should just get along instead of fighting each other. And I think that's another good lesson. But within parasitic fungi, there's sort of two flavors of it. So you have biotrophs where the host lives. Examples of that would be rusts, and you guys are heading into spring. So if you go out and look at blades of grass or rose leaves, you're going to see lots of powdery orange stuff on a lot of these things. Those are rusts. So those are interesting basidio plant parasites that kind of jump different hosts throughout the year, and they produce orange spores and all these different kinds of spores. And those are things floating around in the atmosphere. They don't kill the plant, but they often affect the production. And so this is a big deal for agriculture because a lot of crops get affected by rusts, and they spray with lots of fungicides to treat them. But when you spray with broad-spectrum fungicides, you're also diminishing the microbial diversity of your soil, which has other unforeseen consequences. So another one is smuts. This is another kind of basidio plant pathogen. Something you guys might have heard of would be like corn smut or wheatlicoche. That's an example of a smut. And then there's these things called beetle hangers. I didn't actually get a photo of these in the book, which is why I have in the presentation. And they're sort of like little fungal STDs for beetles. And when beetles like do the deed, they can transfer these things, and they don't go inside of the beetle. They're more just like external and cosmetic. But there's people out there who spend their entire lives studying these tiny little fungal growths that grow on beetles that are like sexually transmitted. I think that's just hilarious and kind of cool. Um, so there's also parasitic fungi that kill their hosts. So examples of that would be like the entomopathogenic fungi, the cordyceps that take over the body of the ant and march it up a branch. The ant is no longer alive. It's full and moving because of the mycelium is moving it, but it's not moving because it's alive. And other examples of this would be tree killers. There's the classic example in America that happened was Cryphonectria, which destroyed all the American chestnuts. And so those are still around and unfortunately still impacting the ability of the American chestnut to come back. Uh, an example of something that can kind of switch between these categories, because a lot of fungi, depending on their form and function, what part their life cycle they're in, can have different ecologies, because fungi are ultimately very opportunistic, and that is, they're always going to do what's best for them. So there are mushrooms called honey mushrooms, and you guys are probably familiar with these. They are tree killers. So they use weaponized mycelium called rhizomorphs, which is kind of like the evil cousin of mycelium. It's all black and forms these tendrils that can like move along the ground, and they sneak up in between the bark and the living tissue of the tree. and they girdle the tree. They cut off the xylem and the phloem and the vascular tissue. So they kill the tree, and then they transition to being saprotrophic and they eat the tree. So this is a great example to now talk about saprotrophs and these decayer fungi. So there's different flavors of this. You have white rot fungi, which are degrading the wood and using really powerful enzymes to degrade the lignin, which is this big complex molecule that cross-links wood and gives it all its structure and allows it to be hard. So white rots figured out how to degrade lignin, and they leave wood with this kind of like shredded texture. So examples of white rot fungi would be something like oyster mushrooms, lion's mane, reishi. Those are all things that are white rots. And then you also see in the forest brown rot fungi. This is a little more common on conifers, and they leave kind of a cube rot. And it's targeting the hemicellulose and leaving behind the lignin. Examples of that would be something like a fomitopsis or even a chicken of the woods or brown rot fungi. Beyond that, you have composters. So those are things that are digesting sort of more broken down substrates. That would be like Agaricus bisporus, a portobello mushroom that we grow commercially. People, if you're a homeowner, you might have dealt with soft rot. That's a kind of a scary kind of like fungal thing that can like degrade the lumber from inside your house. Happens certainly in very like moist areas. And then you have stuff like anaerobic fermentation, which is breaking sugars down into alcohol, and that's what yeast do. So other examples of things that kind of change their what's called ecological guild or their trophic mode. I talked about the yeast that lives in our skin, this Malassezia. So that can be both a positive thing by living on our skin and precluding other bad stuff from landing on us. But if we get an overgrowth of it, then we have dandruff or skin lightning. This is also the case with something like Candida, which is a yeast that lives at all our orifices. But unfortunately, most humans have experienced something where Candida kind of takes over. And I liken this a little bit to like, your body is the club. The Candida are the bouncers at your orifices, keeping the bad guys out. But occasionally the bouncer comes inside the club and starts a fight, and you're like, "Go back outside. We don't want you here." And that's kind of what happens. And there's a little bit of like crosstalk that goes on. I'll talk about it later on. But that's the idea of these dimorphic yeast, things that are able to go from being a yeast form to an invasive filamentous pathogenic form, and that's when they become bad news for us. Plants contain Xylaria or Zarlariaceae, is a whole family of stuff called carbon fungi, and these are living inside of plants as part of their sort of microbiome. They're endophytes inside the plant, and they're usually in an anamorphic mold form when they're inside of the plant. And then when the plant dies, these guys will undergo sexual reproduction and form mushrooms and then start producing spores. And the whole point of the lifestyle that these carbon fungi do is they're living inside of plants so that they're the first on the scene. They have first shot at all of the sugar and the nitrogen that's in the plant as it's dying. And so they have this kind of cool thing where they're like part of the microbiome, and then they're there to like eat it as soon as it's available. So examples of different things that switch their form and function and how they eat based on where they are in their life cycle. Okay, so here's an overview of mycorrhizal relationships, and I think a lot of times people have very positive thoughts and feelings about mycorrhizae. Certainly, if you've seen something like the Fantastic Fungi movie or even Avatar, you can imagine like a big tree with the roots running out underground. You see kind of like the blue signals and the things moving around, and that's a great visual, but it's not necessarily representative of what's actually happening underground. And the thing I want you guys to take away from this is there's two big types of mycorrhizal relationships that you should be aware of, and they're fundamentally very different from each other. Arbuscular mycorrhizae are very, very common. They're associated with about 80 to 90% of the plants on the planet, and they go inside of the plant cell and form a little arbuscule. It's like a kind of a knotted structure that helps them do the nutrient exchange, but they don't produce mushrooms. And they're more associated with like human crops and deciduous trees and lots of agricultural stuff, but they don't form very long networks. So they sink some carbon in the ground, but they don't sink quite as much as the ectomycorrhizal relationships. And these ectomycorrhizae, or ECM, are only associated with that 1 to 2% of the plants on the planet, but it's almost all trees, and especially conifers. So these guys like to wrap the roots in sort of a sheath, and that's how they do their nutrient exchange. They don't actually go inside the cell, they're like sheathing the roots. And so these ectomycorrhizal fungi are the ones that do produce mushrooms. So we care more about them, and they're able to form much longer reaching networks that are more contiguous, but they're sort of overall less common. And around the globe, there is a distribution of ECM versus the AM fungi that is somewhat dependent on climate. And so usually around the equator, you have a higher diversity of the AM fungi. And then up around the poles and big boreal forest, places where there's lots of trees, you will see a higher diversity of the ECM. And this map here has a lot of gray area. And it doesn't mean that there's not fungi in those gray areas. It just means it's undersampled. I actually got to sit at Stanford like a week ago. I was visiting a professor there, and I found out that he was the one who sort of made this map. And I ended up co-opting it and using it for the book. But he's like, yeah, there's all this gray area just because we didn't have data or samples from there. So there's a lot of unknown here in terms of the diversity of fungi and where they are around the world. But I think it is kind of cool to think about the patterns that you see where the AM fungi tend to dominate in stuff like grasslands and agriculture. You get deciduous forests, and trees will have both AM fungi and ECM. And you get them to the higher elevation, like conifer and boreal forest, and those will be mostly ECM. And then in really high elevation areas, particularly in really poor soils, where you have like cranberry bogs and blueberries at elevation, those have a special kind of mycorrhiza called ericoid, which works just a little bit differently. And these all have some role in fixing carbon and keeping it in the ground, but it was really the ECM and the AM fungi are the two main types, and they're different from each other. One produces mushrooms, the ECM do, and then the AM don't. So keep that in mind. Okay, this leads me to thinking about this model called the mycelium economic marketplace. And this is a little bit different from kind of the prevailing ideas of what's called the wood wide web or a common mycelial network. The idea that all the trees are connected by mycorrhizae and sharing nutrients in kind of an altruistic fashion. And I think that that's been overrepresented, and we all feel really good about mycelium and mycorrhizal relationships because we think that the forest is like one big living organism and creature. And in the research that was done, it's been shown that in forests where all the trees are essentially clones of each other, there can be a mycorrhizal network where nutrients are moving around. So they fed some trees radio labeled carbon, and then they were able to show the movement of that carbon into other trees. And that makes sense when all of the trees are related to each other. However, if you have like a mixed forest with lots of different individuals, that may not be the case. All the trees may not be connected and they may not be sharing. It might be more that every single tree is an individual. And just in the way that if all of us watching this presentation went to a financial advisor and asked them for advice, we would get different advice depending on our personal needs, our level of risk, our assets, that kind of thing. We would build a portfolio that made sense for us. So trees are the same way. It's not that they're all blindly sharing with each other. It's that every tree is trying to stack fungi that can help it. So think about this sort of test thought experiment. You have two pine trees. One is in a valley and then one is on a ridge. The one that's living in the valley needs to recruit fungi that can help it grow really quickly because it has all the water and nutrients it needs from the soil, but it's surrounded by competition. So it's going to grow fast. The pine tree that's up on the top of the ridge doesn't have any competition, but it really needs access to any amount of water and nutrients it can get. So these two pine trees are the same species, but they have a different selection of fungi depending on that tree's individual needs. And this gets even more interesting when we start thinking about the types of fungi and their roles. So we have low risk, low reward fungi, and examples of that would be like a rustla or lactarius. Those tend to be kind of like the penny stocks of mycorrhizal relationships. It doesn't carry a lot to cost a lot to carry them, so lots and lots of trees have those. Something that is a high risk, high reward would be exampled by a porcini or Boletus edulis. I learned about this because I ran the SOMA scholarship program for a couple of years, so I'd read all of the applications coming in, and I'm still doing it. And I love to like see the research that's happening. And this was a student who was studying salmon die-offs in the Pacific Northwest. So they were looking at the sort of geospatial like organization of like where mycelium was based on salmon die-offs. So they were looking at stream systems where the salmons come up and they swim to the side and die after they've mated. And the trees right on the edge of the bank are able to absorb all that nitrogen. But that's not where the porcini were. The porcini were a couple hundred feet back from the stream edge, where those trees had Boletus edulis, and it was able to grow long networks out to the edge of the bank and access the nitrogen there. So the porcini was going foraging for the trees and getting nitrogen, but the porcini have a really high carrying cost. They're taking like 5, 6, 7% of the carbon produced by the tree, which is significant. But the porcini is able to go access the nitrogen for that tree, and so that gets really interesting as a forager because you start to think about like, wow, if this tree has this mushroom I want, what is it about the particular situation of this individual, and how do I transpose that to other places where I can actually find the mushrooms that I now want to forage for? So this is a lot more complicated and nuanced of an idea than just two trees kind of shaking hands underground and sharing nutrients, which is originally what I was going to go with. But I didn't want to get stuck in the paradigm of sort of oversimplifying to the point that I'm no longer being truthful. So, well, the idea of like mycelium is the internet of nature is a good gateway concept. It's just not representative of what's actually happening down in the soil. And if you want to learn more about some of this stuff, there's links in my book, but there's also this great paper out of Toby Keir's lab called Mycorrhizal Mycelium as a Globe and Carbon Pool, and that talks to you about like how much carbon is being fixed by AM versus ECM fungi. And then this is a little bit of a scientific smackdown, but there's this paper called Positive Citation Bias and Overinterpreted Results Lead to Misinformation on Common Mycorrhizal Networks. And this is kind of a rejection of that wood wide web idea, basically saying that we don't have enough data to actually say that's really happening. And in a lot of situations, there may be a lot more competition and complexity happening than we necessarily give it credit for. So I like to think of these trees as individuals, and then the fungi are kind of like bargaining with the trees to see if they can get the best deal from the tree. Okay, so that was a lot of the science. I forgot to warn you guys that I was going to spray you with a fire hose of information, and I asked for your consent, but I kind of just got going anyhow. So hopefully you're okay with what I'm talking about. But I love foraging. I am into mushrooms because I am a bella hunter and I like to eat mushrooms. That is why I care about this stuff. In the spring in California, I get to go hunt morels, often burn morels, and sometimes I hunt naturals, which are not associated necessarily with burns. In the summer, California tends to be pretty dry, so I like flying out to East Coast to see my folks, and I forage all sorts of cool stuff there. I've got black trumpets and flame chanterelles and lactifluus and maripilus and honey mushrooms. In the fall here in California, or not here, but in California, I'm in Colorado right now, which is totally different alpine habitat. But in California, if there's early rains, we get a pretty good porcini season. You see all these things sitting on the hood of my car. And in the winter, we get access to stuff called the winter trio. So I have another shot at hunting black trumpets, hedgehogs, and yellowfeet, and occasionally matsutake and chanterelles and romeria and other stuff like that. So I love hunting mushrooms because it is connecting me to the seasons and all of the sort of special food that I get to find in the woods, which is really delicious. But as part of foraging, you have kind of a commitment to your best practices. You got to think about what your impact is on the world, because you don't want to have just an extractive relationship with nature. You want to have something where you're actually giving back, or at least understanding the habitats that you're getting mushrooms from. So these are some of my best practices. I encourage everyone to kind of come up with your own and have your own framework for what you think is best. So one of the first things you should do is look up the local laws, rules, regulations, and try to follow those. Make sure that if you're foraging somewhere, you have permits, or at least permission to be on that land. In general, when you're foraging, it's a good idea to really only take what you need and can reasonably use, or at least share with friends and family. Try not to get greedy, leave some behind for animals and other foragers. There's a lot of ethics that go into this, and there's books that speak to it better than I can. There's Braiding Sweetgrass by Robin Wall Kimmerer. She talks about sort of leave the first, leave the last. And I'm not always hardcore about that. Sometimes I get excited about a mushroom and I just pick it when it's the first one I've seen. But I try to only take the mushrooms that are really prime, and I leave the little ones to grow bigger and disperse spores, and I'll leave the old ones to kind of just spore out and like melt back into the soil. I try to leave at least a third to a half of all the mushrooms I find out in the woods. So the other big thing is be ready to use what you take. You got to go home and not just pick the mushrooms, but actually process them. If you leave them in a bag and they go moldy, you should feel bad, right? You don't want to take something that you could have left in nature and not use it at home. So be ready to actually preserve and cook and eat the mushrooms that you're bringing home. I like to carry my mushrooms around in a sort of open wicker basket. Some people use mesh bags. Whatever it is, it's good to have it something so the spores can kind of disperse, because we as humans can play the role of animals in ecosystems and help move mushrooms around and help them disperse their spores a little bit further than they'd be able to do on their own, because we're mobile and they're not. Learn to recognize invasive plant species and remove them, especially if they're edible. That's a good one. In California, we had the tricornered leek, which is all over the place, and I don't feel bad picking that because it's a wild allium that grows, and I can take as much as I want in the spring, and it's an invasive plant, so more for me. When you're going between different habitats, it's always a good idea to like clean and sanitize the soles of your shoes. So just put a little bit of bleach and some water and dip your shoes in them for a second, and it'll help make sure that you're not transferring stuff from site to site. And when possible, stay on the trails and established paths so you don't compact the forest floor. And if you do go off the path, just be sure to not trample, young shoots and flowers. And I think it's important, especially in the spring when the plants tend to be really small. Help make the forest cleaner when you came in. It's always a good idea to carry a bag for a little bit of trash. I don't like to put like dirty cans and gross things in with my mushrooms, so I keep a separate bag for carrying out, especially glass, plastic, and metal, which do not biodegrade. And one thing that drives me absolutely nuts, so please don't do this if you're a dog owner, but don't bag up your dog poop and then leave it, because dog poop's not great for the environment, but it's a lot worse when you just put it in a plastic bag and let it fester forever. So please don't do that. Whenever possible, try to leave no trace. Treat natural spaces with respect and reverence. And then try to actually lead by example, practice what you preach. So this, I'm telling you guys all this because this is what I'm challenging myself to do, and try to like actually get into in the world. And two, if you're trying to advise people, you don't have to be super hardcore about exactly what it is. It's just have some sort of framework that you're thinking about, and it can be somewhat flexible depending on where you are and what kind of mushrooms you're hunting. But especially when you're talking to other people, you're going to do a lot better if you're nice, right? You catch more flies with honey than if you're really harsh about it. So try to be kind and understanding and empathetic as you're helping people understand how their practices could be better if you see them doing something that's sort of unsustainable, or you think that it could be different. And recognize that human beings are different and diverse. So we're all individuals. Okay, so what are some common mushroom myths that you'll hear? I will tell you the only rule that you can really trust when it comes to mushrooms, and this is stuff people want to hear, like leaves a three, let it be, right? How do I tell what's edible versus toxic? Just give me a rule, give me something I can go with. And the only rule that is universally true for all mushrooms is that there's an exception to every single rule. So none of the rules are good, because most of these rules were designed to help you avoid one particular mushroom, but they've been taken as these general rules instead of like as something that's specifically targeted at a certain type of mushroom. So you'll hear stuff like avoid eating all mushrooms with gills. Well, that's good advice for maybe a very absolute beginner mushroom forager. And what they're trying to say is it's easier to identify polypores and boletes than it is gilled mushrooms, which is absolutely true. But if you don't eat gilled mushrooms, you're missing out on a ton of delicious edible species. So that's kind of silly advice. White gilled mushrooms are deadly poisonous. Well, that's meant to help you avoid the toxic amanitas, but then you'd miss out on something like matsutake or oyster mushrooms, which have white gills and are great. Mushrooms growing in grass are safe to eat. There are lots of great edible mushrooms that grow in grass, but let me introduce you to a mushroom called the vomiter or Chlorophyllum molybdes. This is one of the most common mushrooms that grows in summer in grass in North America, and if you eat it, you vomit. So I wouldn't call that a safe mushroom to eat. Boletes that stain blue are poisonous. I think this is advice that primarily comes from Europe, where they have a few more blue staining boletes that are toxic. Here in North America, especially on the West Coast, we have rubroboletus, which is a stains bolete, and that has red pores and stains blue. I think in the Midwest and the East Coast, you also have Boletus huronensis, which is the false king bolete, which stains blue, and that's how you can differentiate it. But that doesn't mean that all blue staining boletes are toxic. It just means that there's really just one that you got to watch out for. And I'd say the majority of blue staining boletes are actually edible. Not all of them are good, mind you, but they're not toxic. This brings me to do not eat boletes with red pores. That advice is probably meant to help you avoid eating rubroboletus, the Satan's bolete. But there are red pored boletes that stain blue that are edible. So you guys probably see the candy apple bolete or the frost bolete exudoporous. So it has these beautiful red pores and stains blue when you cut it. It has a light lemony flavor. It's delightful. In California, we get this sort of big honkin' boletes that have kind of orangey red pores and stain really dark blue when you cut them. They're called swillelis or liver boletes. And as long as you cook them well, they're fully edible. They're a little bit musty and I don't love them, but they're not toxic. You'll hear this a lot. If animals are eating a mushroom, then it's safe for you to eat. Well, I have a question for you guys. Would you let a squirrel drive your car? Probably not. So why would you let like an animal dictate what mushroom you'd eat? Because if you wouldn't trust them to drive your car, why would you trust them to tell you what mushroom is safe to eat? They could eat a mushroom and then go off and die somewhere. You don't know what's going to happen to that squirrel. So don't trust yourself to an animal. Learn your mushrooms one by one and build up a compendium of knowledge allows you to forage safely instead of just depending on some of these sort of silly rules and antiquated notions that tend to sort of get propagated in amateur circles where people aren't thinking critically. Okay, so if it's small and brown, throw it down. That's pretty good advice for avoiding a lot of toxic mushrooms, in particular Gallorina, which have amatoxins and are deadly toxic. But you'd be missing out on some tasty mushrooms like Marasmiris orates, which is a little meadow mushroom that's pretty good. And in California, we have candy caps, which are beautiful little brown mushrooms that when you dry them out, they smell like maple. And then this is one I think is pretty relevant to you guys. So all polypores that grow in wood are safe to eat. Well, let me introduce you to the nesting polypore, a Haplopilus, which makes a beautiful purple color, and dyers love it. But if you eat it, it can cause like severe brain damage. So not every single polypore that grows on wood is safe to eat. And that's kind of my point in all of this. There's an exception to every single one of these rules. And so you can't trust the things you hear. Instead, you have to learn the actual mushroom itself and then do research to figure out, is this a safe mushroom that I should consume? Okay, one of the biggest things I run to online, particularly as I've been posting mushroom content for 9 years now, and I have a huge amount of content that's out there, is people who have really strong feelings about whether you should cut or you should pick a mushroom and the method of harvest. So there is kind of a prevailing view that somehow cutting a mushroom is going to cause less disturbance to the mycelium below. That seems reasonable, right? There's an alternative viewpoint that cutting a mushroom may actually expose the base to pathogens and other stuff that can now infect the mycelium. So there's kind of these competing viewpoints is like, is cutting or picking better? And all of this happens with very little data. The only long-term study that has been done on cut versus pluck was done in Switzerland, and there was a follow-up study for about 14 years in Norway. And they did a pretty cool experiment where they had three types of plots. So they had unharvested plots, they had plots that were harvested by picking, and they had plots that were harvested by cutting. And this is some simplified data, but this is over a long span of time. And basically, what you see is from year to year, the number of mushrooms and the number of species will go up and down, but they're going up and down as a function of the year. And at no point is the difference between those different treatments significant. So, from a science perspective, you can say there's different amounts of mushrooms every year, but the three different treatments really aren't making a difference in terms of the number of species or even the diversity of species. Or it was also like the size of mushrooms and the number of mushrooms. So what they did see a slight difference was for the plots where they were harvesting mushrooms versus unharvested, not harvesting, they saw slightly more mushrooms in the plots where harvesting was happening. And I think that speaks to our role as animals and moving mushrooms around and helping to spread spores, is we may actually be playing a positive role by going into the woods and messing around with mushrooms because we're helping them spread the spores a little bit further than they would be able to do on their own. Uh, the other thing that was, I think, interesting from this study was they were looking at compaction of the forest floor because sometimes in some of the plots they had kind of boards that people would walk on, like a little like walkway, and then sometimes they were walking on the ground. And where people were walking on the ground, they were causing soil compression. And while it didn't affect the diversity of species, they did see fewer mushrooms in areas where the soil had been compacted. So that speaks a lot to land usage and how much people are walking around in the soil. And this whole idea of like cutting versus plucking, it has nuance. I think ultimately it probably doesn't matter in a general overall sense, but certainly when I'm picking different kinds of mushrooms, I do think about it. So for stuff like chanterelles and porcini, I pick those because there's a significant portion of the stem underground that's edible, and I will trim off the dirt and keep that underground portion of the stem because most of the time I want to take it home and eat it. For something like a polypore like chicken in the woods, maybe the big cluster growing on a tree, and you don't want to rip the whole cluster out of the tree because you want to leave some of that mushroom behind. So what I like to do is take a knife and cut off the kind of growing edges, which tend to be the most tender part anyhow. That's the part that I actually want to eat, and then leave the base of the mushroom in the tree. So that's always a good thought. If you run across a cluster and you want to take the whole cluster, use a knife to cut some off. And it probably doesn't matter whether it's going to like cause disease or it's not. Mushrooms get eaten by animals all the time, and animals have absolutely no regard. They're not walking around with a knife carefully slicing things off. If you've ever seen a bear eat a mushroom, it's just destroyed afterwards. So I don't think that what we do in terms of harvesting practices really matters, and I think people should just be nicer to each other on the internet because I've gotten a tremendous number of death threats over the issue, particularly of cutting versus picking mushrooms, which is very silly. Okay, I like to hunt mushrooms and forage them and bring them home because I like to cook and eat them. That is my main motivation. I am a belly hunter. So one of the things I see with mushrooms is people do the same cooking method all the time. And then a lot of people say, well, I don't like mushrooms because I don't like either the flavor or the texture. And more often than not, it's the texture. And that's because you've been cooking mushrooms wrong your whole life. The way that most Americans are taught to cook mushrooms comes from sort of the French tradition and Julia Child. I love Julia Child. I grew up a couple blocks away from her house in Cambridge, and I've idolized a lot of the work that she's done in her life. But her and the French were dead wrong about how to cook mushrooms. So the prevailing advice for cooking mushrooms is to put a lot of oil and butter in a pan, bring it up to heat, and throw a couple of mushrooms in, but not too many because you don't want to crowd the pan. And then you're supposed to get them very brown. But the problem that happens is mushrooms are kind of like a sponge. So if you put fat into the mushroom, it's not going to be able to squeeze the sponge. What you want to do is squeeze the sponge, get all the moisture out before you add any fat. And the way you do that is by doing what's called a dry saute, or in some cases, a wet saute. Basically, you're putting your mushrooms into a pan with no oil or butter. And this is going to feel weird the first couple times you do it, but this is how you do it. Put them in, add a little bit of salt, and have the heat on and start kind of sweating them out. And for something like chanterelles, they're very wet. They will dump a bunch of liquid and become their own little chanterelle soup within, a couple of minutes. For cultivated mushrooms like an oyster mushroom, they may be pretty dry, and so that's where it's helpful to add like a tablespoon or two of water to kind of help steam the mushroom down and just get it flat against the pan. Once you've cooked that water off, then you can add a little bit of neutral oil, like a veg oil, and a little bit more salt, and you fry it in there 5, 10 minutes on medium low heat. And then at the very end, that's when you take your big knob of butter and some garlic and a little white wine. You kind of like pick them up and get them really like delicious and crispy at the very end. And you can get that perfect brown crispy mushroom even if they started all wet. But when you've done that, you make sure that you've squeezed the sponge and really get that nice meaty chewy mushroom texture without having to be kind of like insipid and floppy and not good. And so you also improve both the texture and the flavor by using this dry/wet saute method. Beyond just doing a pan saute, though, there's so many other ways to cook mushrooms, and I find that people pretty much just do one way of cooking mushrooms. And it's also that mushrooms are often put into a pan kind of plain with a little bit of salt. And you don't normally just cook meat straight with some salt. You often flavor it. You would put a marinade on. So this is where I beseech people to think about treating their mushrooms a little bit more like meat and consider doing the marination step, consider doing like a pre-cook step, and then like another cooking step to really emphasize the flavor and texture of a particular mushroom. And so that's why I put in this infographic to kind of like think about all the different ways that you could have mushrooms. So I had a dinner the other night where I watched a chef very plainly roast a bunch of morels, a little bit of butter and some thyme. And I said, well, that's great, but I'm a little worried that like the morels are going to taste kind of plain because that's not he's not putting that much effort into cooking it. And then he surprised me because he took the morels after they'd been roasted and he put them on the grill and then he charred them. So he added a nice smoky flavor and then he put them into a polenta. So really there's like three different cooking steps at play there. And it really emphasized the flavor of the morel as well as improving the texture in the final dish. So I love that people think about how to kind of combine all those different elements into something. And I encourage you guys to like get creative in the kitchen and think about all the different ways you can use mushrooms in different ways. Maybe in the same mushroom, we have like some that's been sauteed and then some that's been fried and some that's been smoked. And you bring in a lot of different like textures and flavors to your dish. Beyond just cooking them, I also really like to preserve them. So in general, I dry a lot of my mushrooms, and for certain mushrooms that can really enhance the umami flavor of the mushroom, which is a great way to kind of like preserve them while making them more flavorful. But some mushrooms, when you dry them out, kind of lose all their texture. So something like a chanterelle, I don't really like dried chanterelles because it doesn't do much to improve the flavor, and it kind of ruins the texture. So what I like to do in that case is that we'll cook them with that dry saute method and freeze them. And I try to make the amount of oil and butter I use when freezing them very minimal because fat is what picks up some of those weird like freezer burn characteristics. So if you either boil them or do a wet dry saute and then freeze them in little usable packages, that's a great way to kind of preserve the texture of a mushroom. Beyond that, though, there's a lot of different ways to preserve mushrooms because you can do canning and pickling. You can do a conserva, which is kind of like a quick pickle. You like boil the mushrooms in a little bit of like a light brine vinegar, and then you top them off with oil, and they keep them in the fridge. And you can kind of pull them out and eat them as snacks, or put them on pizza, salad, stuff like that. I really like to make syrups and then candy mushrooms as well. So preserving with sugar is something you don't necessarily think about with a mushroom, but it can be really great. Last fall, I was cooking chanterelles down in kind of a caramel and then mixing them into like an apple pie filling, which was really phenomenal. It's great in like apple pie or in a turnover. You can also do liquid infusion. So with candy cat mushrooms that smell like maple, I'll put those in whiskey. I've had truffles preserved in vodka. That was a great way to kind of keep that smell. It was something like matsutake, which doesn't dry well, nor does it necessarily freeze well because you don't get the aroma of the matsutake and the frozen stuff. I like to preserve those in sake, and I'll add a little extra salt and sugar and MSG to make kind of like a matsutake mirin almost, and then I'll save that in the fridge and pour it into dishes and cook with it throughout the year. So those are just some different ways you can cook with and preserve mushrooms, which I think is pretty great. And all of this is following the flow of my book. So we were in the foraging chapter, now we're in the cooking chapter, and now we're going into the fungal intelligence chapter and some biochemistry stuff. So I think fungal intelligence is vastly underrated and underappreciated Because fungi don't have brains, they don't have eyes, they don't have noses, they don't have hands, feet, fingers. There's nothing about them that makes us kind of identify with them. They have a completely different type of intelligence that is not based on animal cognition or consciousness. Instead, it's what's called network intelligence. And that's the ability to coordinate lots of decisions across a diffuse network. So every single little growing mycelial tip is its own individual, but it can decide to go up or go down, go away from stress or towards food. So each of those mycelial tips is making decisions. And yet, if the mycelium is able to sense like the nitrogen from those salmon, they can grow towards it and they can cut off all of the other growth and put all of their effort towards growing towards food. Because unlike humans, they're not individualistic. So I don't know if you guys ever try to get like 10 people to go out to dinner at the same time. People are like, well, I want burgers, I want pizza. Uh, Humans are far too individualistic to actually make those decisions, but fungi have this amazing capacity to coordinate the decisions with network intelligence. So, some of the things that I think make fungi very intelligent would be their ability to sort of sort, forage, and search for resources. I talked about them being able to go get the nitrogen from the salmon die-offs. It's also been shown that if you disrupt mycelium as it's growing towards food, if you like ran a car across the mycelium and disrupted the soil, it would then be able to keep growing in the same direction. So it remembers where the food was. It doesn't have to search all over again. It knows where it is. It can sense the environment and chemical cues because it can sense like where the dying salmon are. So it can kind of smell and taste a little bit. It's able to do long distance communication and coordination, even active transport across a mycelial network. That's part of how it's coordinating its behavior. They can learn, they can adapt, they can anticipate future events, learning from past events. They can do animal manipulation. So we talked about the cordyceps and how it's able to like locomote an ant up a branch and grow a mushroom out of its head. Another great example of this idea of what's called the extended phenotype, something exerting its influence on the world well beyond its actual body, would be alcohol. And if anyone here has ever enjoyed an alcoholic beverage, you are experiencing the extended phenotype of yeast. It wasn't your fault you did something dumb while you were drunk. It was the yeast's fault. I'll stick with that one. Um, you have ability for fungi to facilitate plant-to-plant interactions, at least in some limited context. It may not always happen across like a whole ecosystem, but certainly like there's been stuff demonstrated like two tomato plants beside each other, one of them could get infected and it'll tell the other plant beside it through the mycelium that there's a pathogen nearby, so hey, you better activate your defenses. Fungi can recognize self versus non-self. So when we think about the idea of mycelium down in the soil, it may not all be like hunky dory and things getting along. It might be that things are in competition. Again, symbiosis is both positive and negative, and there can be fungi, mycelium particularly, that doesn't get along. And that's where they can undergo some competition and even chemical warfare. And you can put two different types of mycelium on a plate and watch them kind of battle it out with each other. And there might even be like a third kind of mycelium that's hanging back, waiting for these guys to battle it out, and then it'll come clean up when everything else is kind of resolved. And fungi also have the capacity to sense light day length via photoreceptors in their cell membrane. So there's a lot of these behaviors that fungi have that are considered intelligent, but sort of different from animal intelligence. And I think it's important to kind of recognize that. And it's really interesting as we start to think about how do we understand fungi and all of the cool things that they do in the world, just recognize that while they don't have brains, they kind of are almost like a living brain unto themselves. Okay, there's a surprising role for fungi in the human body. We talked a little bit about like the Malassezia yeast that's on our skin. We talked about some of the candidas. Here's kind of a mechanistic thing where fungi are communicating with each other in a process known as quorum sensing. So this is what happens when you have the bouncer at the club, your candida yeast at all your orifices, and you get some sort of change in hormone levels or in cosmetic products or your stress, whatever environment is changing, and they kind of get together until they reach a certain point that enough of this tyrosol has been produced through their metabolism. They say, you know what? Now we're going to turn into this invasive filamentous form, and you're going to get all red and itchy. That's what a yeast infection is. And this isn't, very dependent on these sort of environmental factors and like various like physiological factors that you have going on in your body. But beyond candida, there's fungi present in a lot of our tissues and all over our body. And the way that fungal diseases usually manifest on the human body is either as a skin rash, which is fairly easy to treat and usually not a big deal, but it can take a while to get established and also a while to get rid of. You can get respiratory infections, which is more serious, and that's when you get like mycelium and stuff growing in your lungs, and that can really impact your breathing. And then there's the third most serious kind would be like a sepsis or like a blood infection. You actually have like literal clots of mycelium going through your blood vessels, and that's very bad. That doesn't happen usually unless someone is undergoing chemotherapy or is completely compromised immune system. But a lot of these fungi in the human body that can cause problems are there all the time. They're just fairly well controlled for by our immune system. And so that's why when you're undergoing chemotherapy, or you have something that really knocks back your immune system, then these fungal diseases become a bigger deal. Okay, so I talked about some of the ways that fungi can hurt us. Let's talk about some of the ways that fungi can help us. One of the big things I see is that fungi can make really good, sustainable food. They don't take a whole lot of land, they don't take a whole lot of power, nor do they take a whole lot of water. So fungi produce really high quality protein because fungi are more similar to animals than they are to plants. The kinds of amino acid that are in fungi are basically a one-to-one match for what we need, whereas plants skew heavily to giving you like far too much lysine. So if all you eat is soy, you're going to end up with like weird health issues. But fungi have really good protein that's good for us. It's nowhere near as protein dense as something like meat, but it uses way less water. And especially in a place like California, where water is at a premium, I think it's important to consider like, well, where is our protein coming from? Because almonds and beef are extremely water intensive, but something like mushrooms is very, efficient and also uses waste products. So in that sense, I think they're really sustainable and good for us. I get asked a lot question about what mushroom is sort of best to eat health wise, and I say, well, eating lots of different kinds of mushrooms is probably going to be the best, and trying lots of mushrooms can help open your mind to lots of delicious possibilities. And one thing I like to kind of point out to people, if someone eats an eggplant, doesn't like an eggplant, does that mean you hate all vegetables? No, you just might not like eggplant. So try other vegetables. So if you've eaten a mushroom and you don't like it, well, try other types of mushrooms. They do taste different. They have different textures. So be a little experimental. In terms of the nutritional content, mushrooms can be different from each other. They have certain different amounts of protein and calories and things like that, but they're all relatively similar. They're all sort of in the same range. And the really important thing about eating different kinds of mushrooms isn't necessarily the nutrition, it's the fiber. And to me, that is the most important nutritional benefit of mushrooms is the complex dietary fiber that they can contain, because that fiber can act as prebiotic food for our guts. So there's lots of complex linkages in the polysaccharides in mushrooms, and those can be broken down by specific types of bacteria in our gut, which lead to more microbial diversity and then an overall better immune response and better cognitive function and better, inflammation response, all this kind of stuff. So eating a diversity of mushrooms is really good for you. Highly recommend that. I talk about in the book medicinal mushrooms, but I'm not talk about them too much in this talk because I think they're kind of overrepresented as like marketing hype most of the time. So I like to instead draw people's attention to the actual drugs and therapies that we have created from using fungi, because fungi are nature's chemists. And a lot of the times we look to nature for solutions to our problems. So great examples of some really concrete things that we have gotten from fungi would be penicillin, probably the most important antibiotic of all time. That came from the penicillium mold. So penicillium mold is the blue stuff in blue cheese. It's the white stuff on the outside of your brie, and it's even the white powder on the outside of your salami. That's all penicillium mold. There's a very famous mushroom personality out there who tells everybody that his mother's breast cancer was cured by taking turkey tails. And while the turkey tail probably helped support her immune system while she was undergoing chemotherapy, this is the actual chemotherapeutic drug that she was taking. And this is a drug that was originally isolated from fungal endophytes living inside the Pacific yew tree. So I think that's really cool because it was a fungal derived chemotherapeutic agent that actually saved her life and helped beat the cancer. And the turkey tail probably helped support her immune system as she was undergoing that chemotherapy. You may know someone who's taking statins, and those are to help reduce cholesterol and low density lipoproteins. That was originally isolated from Aspergillus fungi, which is related to koji, the stuff that makes miso and soy sauce. And then we have something called cyclosporin, which is an immune suppressant. Right, if you know anyone who has an organ transplant, they're probably taking cyclosporin every day so they don't reject their kidney. But cyclosporin was originally isolated and discovered in Tollypocladium, which are these interesting asco kind of cordyceps-like mushrooms that infect Elaphomyces or deer truffles. So the Tollypocladium uses this very complex molecule to suppress the immune system of the truffle that's infecting and then grow a mushroom. And we have taken advantage of that chemistry to make sure that we don't, reject a kidney after getting an organ transplant. So I think that's all pretty cool, interesting stuff. Okay, so I talked about how fungi can help us, but how can we help fungi? What can we do to help them tackle the issues in our world? And a lot of the problems with mushroom hunting aren't caused by your local forager. It's not that somebody in your area picked too many mushrooms. It's that we have catastrophic climate change happening all over the place and things are completely out of whack. And it just keeps happening. The hits keep coming. We have issues with invasive species, pathogens, development, land usage, degradation, agriculture, flooding, pollution, wildfire. All these things are way bigger than the individual. We as human beings struggle to deal with these things because we can't go out and solve these problems on our own. And it feels bad to see someone take too many mushrooms or do something unsustainably. But even ultimately, no matter how bad one person is, it's not as bad as like the mass action of like governments and corporations and the things that are like causing the systemic causes of climate change. But we can't fight those as individuals. So what can we do? Because shaking your fist at the man doesn't necessarily solve the problem. We can come together and support organizations like the Fungi Foundation, which was founded by Juliana Furci. She's an amazing Chilean mycologist, and she started the Fungi Foundation with the goal of helping sort of be a voice and an advocate for fungal conservation. And they have this thing called the 3F initiative, which I think is really smart, and it's the flora, fauna, and funga proposal, which they are taking to governments around the world and asking them to adopt the term funga into their conservation legislation language, such that if you're doing an environmental survey of an area before you like mow down a forest and put it in a parking lot, you actually should go there and see what organisms are growing there and observe not just the plants and the animals, but also the fungi. And that way, say if you have like a state mushroom, you can fight against a developer who's trying to mow down your local patch of forest by saying, well, hey, our state mushroom grows here. Don't we want to preserve that? It gives people a little bit more power in terms of what they're able to do when you start integrating stuff from a top-down approach instead of just it being bottom up. So we can work bottom up and do our grassroots stuff, but I think supporting these bigger organizations that are able to go advocate and lobby for fungi in a big picture is something that we all can support and something we can do meaningfully also as mycology clubs. Um, there's the idea of mycoremediation, and this is where it's not as simple as taking like a bag of oyster mushroom mycelium and putting it on an oil spill. That doesn't work. It's been debunked. It's not useful. What is useful is going to a polluted or contaminated site, sampling some of the microbial diversity that's there, and then taking that stuff back to a lab, doing some isolation, some screening, trying to figure out what grows best on the pollution, and then maybe doing some selection and optimization and growing up a bunch of that organism and taking it back out into the environment and reintroducing it and allowing the fungi to serve as like sort of the first step in this remediation of toxic compounds. It doesn't mean putting the fungi there is going to solve everything, but because fungi are excreting enzymes and changing the world around them, they can be the first step in a series of ecological steps and successions that allow for remediation of that pollution. And that's one way that we can kind of help fungi along and making things happen. Okay, so humans are really good at making lots of production, but we also make lots of waste. And one of the things that you look at nature, well, nature's not quite as productive as human systems, but there's very little waste because almost everything gets shunted into something else, is digested and eaten somewhere in nature. And so when we look at human practices, we could think about all the different ways that we can integrate fungi into what we do to make us more sustainable. So, in terms of producing medicines and food and mycomaterials and enzymes and agriculture, there's so much we can do by taking our waste product. Ooh, excuse me, taking our waste products and integrating fungi as a way to kind of reduce the waste or transform it into something else that has a valuable added product. Uh, a great example I like to bring up here, and I think it's really interesting because you don't think about this, but the detergent you use to clean your clothes, the reason that Tide is so good at getting rid of stains is because they grow up giant vats of fungi and extract enzyme from them. And they've been doing this for like 50 years. But if we started thinking more and more about how we could use fungi as tools and how we could help our kind of like reliance on degrading waste and allowing them to kind of do what they naturally do and feed our waste streams into fungi, we could be getting, new medicines, new materials, new production for agriculture, even new forms of energy by simply integrating fungi. And that's the idea of this mushroom circular economy. Okay. Part of that is growing mushrooms. And so I think mushroom cultivation is fascinating. I think it deserves an entire book. So in my book, there is an infographic, but there's not a lot more on it because mushroom cultivation is so different that it's like it deserves its own book. But here's sort of a basic flow. You can start out by cloning a mushroom or getting some spores, turning it into pure culture on a plate, then using that mycelium to inoculate some grain. And once you have some inoculated spawn, you can do all sorts of different ways of cultivating mushrooms, from tray cultures to bags to jars to columns and mounds and stumps and logs, all sorts of stuff. I do a bunch of these things in my backyard, and it's really fun and gratifying to grow your own mushrooms. And you can even do it with waste from your own yard, which is also very cool. Okay, so now we're getting into like the mushroom part of the book. One of the things I see is every time you see a mushroom book, mushrooms are represented by the anatomy of an amanita. So amanitas are a good sort of blueprint for what a lot of mushrooms look like, but they have some features that many other mushrooms don't have. Most mushrooms don't have little warts or dots on the top. Those are a function of the universal veil on amanitas. Lots of mushrooms don't have a ring, an annulus, or a skirt on them, and most mushrooms don't have a sac on the bottom called the vulva. So while amanitas are pretty iconic, they don't necessarily need to be the poster child for all mushrooms because there's many other different forms and types that you're going to see out there. So I included stuff like polypores, gastroids, where the spores are on the inside, and then for the ascos, you sort of have like two fundamental things that are either cups, where they're lined with the asci, or you like the xylaria, you have these carbon fungi where there's little dots called parathecia, and all those little dots are filled with the asci inside. So I'm just trying to give you a few different looks at what mushrooms can appear as. And that's because in the very center of my book on page 145, there is this diagram on the right, which is the 16 morphological archetypes that I think you can pretty much take any kind of mushroom and fit it into one of these categories. And then in my book, you can go read through all the different genre names that correspond to these different categories. And these are not genetic categories, these are morphological categories. So they're based on the way that things look. And this is really important to understand because there's lots of mushrooms that have evolved into the same form and function, but come from different genetic backgrounds. So all tooth mushrooms have little teeth, but they're not all genetically related. And the same thing with gilled mushrooms. There's lots of things that have evolved gills, but they're not all coming from one common background. And there's things that have a common background, but have diverged into two totally different morphologies. And so on the inside cover of my book, I have a big taxonomic table, and I invite people to kind of compare and contrast both morphology and taxonomy because those are two things that they're sometimes in opposition to each other. Sometimes they make sense, but sometimes they don't. And so what I'm really trying to help you guys do is just to recognize mushrooms and get a sense of kind of how to bin them quickly as the overarching structure to understand what to do with your mushrooms. And in that sense, I see my book as like a guidebook to other guidebooks, and that's why it's called Passport to Kingdom of Fungi, because it's your entry into the world of fungi. And I don't like dichotomous keys because I've never really used one successfully, but this is sort of the same idea of you can ask, like, are you above ground, below ground, are you an asco or a basidio? Like, what category of mushroom are you ending up in morphologically? Okay, then we get into edible stuff. And I get the question a lot on hikes, and people say, is this edible? Yes, no. Well, give me an answer. And I'm like, well, it's a little bit more complicated than a yes, no. There's a spectrum of desirability when it comes to edibility. And in a lot of books, the term choice has been thrown around for a long time. And it's confusing, especially for beginner hunters, because they're like, I don't know what choice means. What would I choose? And experts will say, well, you choose to eat this. I choose this one because it's better. And it's like, but beginners are like, I don't know what I like, so I don't know what to choose. And it's just kind of an overall confusing thing. So I went with what I called the 5-star system. I think this is inherently pretty easy to understand because if I ask you if you want to stay at a 5-star motel or a 1-star motel, I think you know the answer to that. So when it comes to edible mushrooms, I like to stick with the 5, 4, and 3-star, the incredible, great, and good. Uh, I very intentionally rated chanterelles and porcini as 4-star mushrooms in my book because I kind of want to upset people. I like chanterelles and porcini, but I don't think they're incredible. I think they're great. And I am telling people that very, very much these are my subjective ratings. So at some point, I hope someone comes up to me all blusteringly angry and says, "Chanterelles are my favorite. How dare you?" And then we can have a great conversation about how they cook them and what their favorite things are and that kind of thing. Um, Generally, a lot of mushrooms are going to fall into the one-star category. So out of, let's say, 150,000 species of mushrooms, there's probably like 3 or 4% that are really great edibles. And then there's probably a few percentages that are toxic, but almost everything in between is just going to be like a 1 to 2 star mushroom. Like it's not going to be toxic, but it might be unpalatable or bitter or just tough. It's not going to be stuff that you necessarily want to eat. Speaking of that, I also added another category called special preparation or medicinal. Example would be something like turkey tail, which uh, is not toxic. But if you've ever tried to chew on a turkey tail, you can chew on it for hours. You're not going to get anywhere. They're really tough and wiry. You can especially prepare them into like a powder or a tincture or a tea, but you can't just eat one straight up. And if you can, good for you. But I'm not going to call it edible. I consider it sort of a special preparation. You can also take some toxic mushrooms and boil the toxins out of them to turn them into an edible mushroom. So I wanted to have an extra category to add that little bit of nuance. Within the book, I have the edible sections organized by cultivated, edible polypores, edible boletes, easy to identify edibles, sort of intermediate and advanced. And the whole idea was to present things where for the beginners, I would recommend sticking to the edible polypores, boletes, and easy edibles. That's where you can start. And as you kind of level up in your mushroom hunting knowledge and you gain more confidence and experience, you can start hunting the intermediates and eventually the advanced. But the advanced ones have a deadly toxic chance of eating something bad. So you want to be absolutely sure you know what you're doing. You don't want to start running before you learn how to crawl, right? And so all of these examples I start with usually at about 5 write-ups for each section. And then there's a whole list of what are called honorable mentions. I would give the picture and a name, but I don't necessarily have room to do a whole write-up. But I want you guys to know what else I would put into these categories. Okay, when it comes to the toxic mushrooms, similar idea. There's a spectrum of threat. So there's things that are deadly toxic, like the death cap in California caused a lot of issues this year because it has amatoxins. And amatoxins are little heat-stable cyclic peptides. They get into your liver and shut down liver function, and then you basically melt from the inside out. It's a very painful way to die. So you do not want to eat those, and they're very dangerous. You can't specially prepare them to make them safe. You have stuff like gyrometria or paragyrometria, which contains hydrazines, which is a volatile toxin that can affect your central nervous system. That's a scary one. People die from that every year when they think they're hunting morels or something like that. You have Gallorhina, which also contain amatoxins, and this is something that people normally wouldn't eat a small brown mushroom, but if you're hunting for like psilocybe mushrooms, you could make a deadly mistake. So you want to be aware of these things. When it comes to dangerous mushrooms, my example is something like the jack-o'-lantern mushroom, which is one that people frequently confuse for something like a chanterelle. And chanterelles are great edible mushrooms, but I think they look very different from jack-o'-lanterns. But if you're a beginner and you're just reading the description of a chanterelle versus a jack-o'-lantern, it is possible to make a mistake. And you should always look at things side by side. But sometimes people get excited and their excitement gets the best of them. They end up cooking and eating this toxic mushroom. And it's not great. It makes you have really bad diarrhea and throw up, and it usually resolves within like 24 to 72 hours. It can be very unpleasant and potentially dangerous experience if you become dangerously dehydrated during that time. There's stuff like the sulfur tuft, which is actually fairly toxic. People don't normally get poisoned by it because it's so intensely bitter that they don't eat it, but it is quite toxic. And there's something here like the Paxillus or roll rim, which used to be consumed as an edible mushroom in Europe until a couple of European mycologists keeled over and died. As it turns out that there's a thing in the Paxillus that over long-term exposure can cause sort of an inherent blood allergy, like an autoimmune blood allergy, where your body starts attacking your red blood cells, and that can be fatal. So these are things that can be dangerous, even though one exposure might not necessarily be like life-ending. And then we have stuff that is in the beware/exercise caution category. So example of this is Amanita muscaria. That's the fly agaric, the red and white Mario mushroom. That's probably the most iconic, recognizable mushroom in the world. It is both edible, toxic, and trippy all at the same time. If you eat it raw, you're going to have a really bad night of throwing up and having diarrhea. Not fun. If you were to boil it a couple times in successive changes of water, you could dilute out all of the toxin that's in there and then eat it safely like another mushroom. And if you were to take the mushroom and dry it out, you would start decarboxylating the toxic ibotenic acid into muscimol. And then if you take the dried mushroom and boil it down with some lemon juice to make a tea, you finish the decarboxylation. And muscimol is a GABA agonist that feels a lot like alcohol. So drinking like a cup of Ambien muscaria tea can feel a little bit like having a nightcap, and you feel kind of like sleepy and a little woozy and maybe a little like loose and happy and fun. Were you to drink a whole lot of that tea, you would suddenly feel like you have the spins. So this is not like psilocybin. It's a fundamentally a different molecule, a different set of brain receptors, and it can be dangerous if you combine it with alcohol and other things. So you want to be careful, but it is a thing that a lot of people are exploring for pain treatment and insomnia, stuff like that. We have mushrooms like scleroderma, or what's known as the pigskin puffball. So all of the books in North America, including my own, will tell you that this is a toxic mushroom. But I've learned through the internet because I had a video of me cutting up and tapping a earthball scleroderma, and it got 40 million views. And I'm like, what's going on? I'm like looking at the demographics, trying to understand because a lot of people watching this video are from Thailand, Vietnam, Philippines, and there's a whole bunch of chatter going on, but I can't understand it because I don't speak these languages. So I was like, hey, can anyone explain to me like what's going on here? Why is this video so popular? And I was told by someone that in the Philippines, scleroderma is their favorite edible mushroom. And I was like, how do you treat it? How do you, this is toxic. Why do you guys eat this? And he said, well, we treat it like a puffball. We only pick it when it's super firm and it's all white inside, and we peel off the outer skin and then we cook it up. And it turns out the scleroderma are in Boletaceae, so they're related to boletes. And when they're all white inside, you can eat them. And I did this last summer. I found one that was like perfectly white and really firm. And I was like, okay, shoot, I'll try this out. So I peeled off the outside, tried a little bit, and then ate the rest the next day. But I really enjoyed it. It was a good mushroom. I wouldn't do it with anything that was changing at all slightly inside of the color. But it's the same rules of puffball, right? You might actually be able to eat this thing as long as you don't eat it when it's more mature. So then you also have like the yellow staining agarics, and those are David Royer calls the lose your lunch bunch. So generally, if you have these sort of phenol yellow staining agarics, they're not good to eat, and you could feel pretty bad after eating them. But they're not deadly toxic. You're just going to throw up, and that's generally the type of poisoning that happens with mushrooms is GI upset, where you have like tummy rumbles or diarrhea or throwing up, and it's only some of the more dangerous and deadly things where you have more severe symptoms associated with them. So I really wanted to have like a nice, easy to understand infographic that had some of the mushrooms, some of the toxins. But as I got into writing about this, I realized that there was like way more mushroom toxins than we even know about. There's a lot of toxins we don't know. And so what I try to do was give examples of these different categories along with their kind of respective threat level that they posed, and then some ideas on treatment and what you should do if you're experiencing some of these different symptoms. So this is my kind of treatment of that in terms of biochemistry of toxic mushrooms. But in the book, I want to introduce you to a lot more than just toxic and edible. So I also talk about medicinal mushrooms. I talk about entheogenic mushrooms, which is sort of a polite way of saying psychedelic mushrooms. I talk about the urban gastroid mushrooms, some of the things you'll see growing around in wood chip beds and in your yard. I talk about dye mushrooms that you can make beautiful pigments and dye fabrics with. I talk about really colorful mushrooms and some of the cool chemistry that's going on there. In a lot of cases, the colorful mushrooms actually are not toxic. You would assume the more colorful they are, the more toxic they are, but that's not true. That doesn't follow with fungi. And I have a whole section on non-mushroom fungi where I talk about rusts, like I mentioned earlier, and entomopathogenic fungi and yeast that ferment our food and molds that make cheeses, all sorts of stuff like that. So the book finishes off with a fungal phenomenon section. My favorite example of fungal phenomenon is guttation. It's basically when mushrooms are growing so quickly they start sweating. This is the Hydnellum peckii, or devil's tooth. And Hydnellum in particular have really impressive guttation when they're young. They get these droplets all over them. They look totally surreal. You would assume this is toxic. It's not. It's just bitter. And it kind of stains your hands kind of reddy orange if you touch it. Some mushrooms are fluorescent, and so this is when you shine a UV light on a mushroom. The light excites electrons in the mushroom. When those electrons come down in energy state, they put off a wavelength at a different light. So you put light in, get light out. And fluorescence doesn't necessarily mean anything about the mushroom in terms of its biology, but it's a great tool for identification, and sometimes it's just really pretty. So it's fun to go around at like a foray and shine UV lights on stuff and see what things look like. My friend even showed me yesterday. He was like in Sweden. He was hunting at night, and they were like, "Oh, it's kind of done for the day. We're going to find anything." And then they shone their UV light on the ground, and suddenly all these hedgehogs popped out of them that they didn't see otherwise. So it can be a really great hunting tool too. Uh, there's mushrooms that are bioluminescent. So the jack-o'-lantern mushroom I mentioned earlier, that's deadly, not that's dangerously toxic, not deadly toxic. Those will glow green, and the bioluminescence in those is similar in its mechanism to the way that like jellyfish or fruit or like the fireflies work. It's an enzymatic system where there's luciferin and luciferase, and they're sort of low level putting out light all the time, but you can't see it unless you're in like a fully dark context. So with some of these jack-o'-lanterns, you're going to want to take them into a closet and sit there, play like 7 minutes in heaven with the mushroom, and just stare at it in the dark until it glows green. I know in California we can see them with the naked eye. I'm less sure of the ones you guys have in the Midwest, whether or not you can see those, but you probably know, and you guys might be able to figure that out as a club. I do know that I've seen Pinella stipicus glowing in real time in the Midwest, so I know you guys have that. Hopefully, you guys have seen a fairy circle before. And I hate to tell you, it's not a portal to the fairy realm or the fey dimension. It's just a big patch of mycelium growing in grass, and there's mushrooms around the outside. Usually, in the middle of a fairy circle, you often see super green grass that's growing because the mycelium there in the middle has kind of decayed and returned those nutrients to the soil. You have examples of stuff like gravitropism, where if a tree is growing and there's a polypore on it, it's growing like this, and then the tree falls over, and all the new growth on the polypore is now perpendicular and pointed toward the ground because spores always like to fall down with gravity. So this is an example of a mushroom that was growing and then it changed orientation. All new growth is now pointed down. I told you that you know, pores and gills and spores like to orient down. Well, sometimes mushrooms will grow with their gills on top. They'll have gills on the bottom, but they'll also grow gills on top because they're kind of confused. We're not really sure why they do this, but it's called rose combing, sometimes referred to as a mutation. And in really extreme examples, you can actually get a mushroom forming on a mushroom, like a double decker, and even seen like a triple decker mushroom sometimes. Uh, you have examples of stuff like rhizomorphs. So that's the evil cousin of mycelium I was talking about. That it's all melanized and it's really like thick cords. That's what honey mushrooms use to creep up and kill trees. But lots of different kinds of mushrooms have rhizomorphs they use to kind of like move around in more stressful situations, and they're not inherently like that kind of fine white mycelium in the soil. So there's different flavors of mycelium. That's what rhizomorphs are. And then this thing here, this big black lump, is a sclerotium. This specifically is chaga, and this is kind of like a fungal battery. It's a sterile conch, so it doesn't produce spores. It's not a mushroom, but it's banking lots of energy in this structure that's melanized to help it protect it from UV, and it's saving energy for later to form mushrooms later on. And this is a really important kind of structure to think about when we think about morels. I know you guys are heading to your morel season, and this is a really important understanding that morel mushrooms fruit from the sclerotia. So for morels to form, their mycelium has to form this little stress structure called a sclerotia, and then that's where the morels are actually fruiting out of in the spring. So I've heard in certain habitats, if you find like a honey hole, it might be that a bunch of that sclerotia somehow got washed downstream and washed up on a bank, and that's why a certain embankment has like all of the morels you've ever seen, and then elsewhere they're not really there. It's because the sclerotia physically got removed to a certain point, and that's where the mushrooms are forming. So that's all kind of cool stuff. Um, Big question is, why do mushrooms smell? So a lot of mushrooms have some sort of aroma, and it may help with attracting animals. An extreme example of this is truffles. And I'm going to tell you that truffles are not a genetic category, they're more of a lifestyle. So every major mushroom family has a truffle form because environments where it's dry, like California and Colorado, it may behoove you to actually live underground where you're not as dependent on like the wet, dry cycles. The problem is if you live underground, how do you disperse your spores? Well, universally, mushrooms have figured out that if you get really stinky in maturity, an animal will come dig you up and help you spread your spores. And that's the whole truffle lifestyle. So people typically think of pigs as being the animals that harvest truffles. Turns out that pigs like to eat fingers, or at least not if not like they end up eating them because they are pigs. So most commercial mushroom hunting and truffle hunting is done with dogs these days because dogs are a little bit easier to train and easier to get along with. But in nature, the animals that are doing the majority of spreading truffles around, and this is not all truffles are good, right? There's some truffles that are beautiful shaved over pasta, and then some truffles smell like burnt rubber, and you would not want to eat them. But again, it's stinky enough that some animal will want to eat them, even if it's not us. So the majority of the animals that spread those spores around are going to be stuff like rodents, like squirrels, rats, wood rats, birds, and then specifically insects are really what's getting down to the soil and spreading the spores of those truffles. There are some really enticing and amazing smelling mushrooms out there. In California, we have the candy cap, and this is a mushroom that smells like maple when it's dried out. So maple syrup contains a compound called sodalon. It's a lactone. Lactones are created by toasting wood. And the classic one that you would all know is vanillin. That is the smell of vanilla. So when you age whiskey in a barrel, you age wine, you're adding the lactones to the wood, kind of softening the beverage, and you get that same kind of softening from soda and candy caps. So I love sticking my candy caps in whiskey, and you kind of bring a little extra maple aroma. And I just think it's really cool that the same compound that's in this mushroom is the same compound that's in maple syrup. It's also in sherry and fenugreek. So those are all of those things have the compound soda in common. Uh, in California, we have a mushroom called the Prince. I'm sure you guys have a version of this where you live. That is kind of an almond-smelling agaric. So it's like a portobello, except this is kind of like marzipan-like character. And that's because it literally contains benzaldehyde, which is what they use to make almond extract. And then you have stuff like matsutake, which is this sort of like cinnamon, seafood, like high note chemical floral thing going on. It can be like a little gym sake or a little more pleasant, depending on where you are and what your perceptions are. But these are all enticing smelling for sure. There's some really interesting smelling mushrooms. So the bluets, to me, smell kind of like a frozen orange juice concentrate. If you've had cafeteria orange juice, you know what I'm talking about. Or if you can say generously, they smell kind of floral like lilacs, but it really depends on what your perceptions are and where those mushrooms are growing. In California, we have this amazing mushroom called the cedar waxy cap, which is this incredible, like terpenic, piney smell. Sometimes it's called Russian leather. That's like a perfume term to describe that smell. And then you have stuff like the blue green anise mushroom and the Clitospia odorata, and that has a uh, anise and fennel smell, and that's a lot like what oyster mushrooms smell like too. So smell can be a great tool for identifying mushrooms, but there's also some really gross smelling mushrooms. So we have things like the sulfur night. This is one that smells like burnt rubber and petrol and mothballs. It's a really nasty mushroom, but like I remember finding it and being utterly just like repulsed by it. Then I somehow kept like going back to it, and be like, oh, it's terrible, but I just want to smell a little bit more. It's human nature sometimes to smell something gross again and again. Um, by far, one of the grossest and smelliest mushrooms I've ever found is this little thing called the stinky squid or pseudocolus. I've seen this on the East Coast, and every time I found it, I swear to God, I found a dead body. And I'm like looking around for like a decaying deer corpse or like a God forbid a human corpse. And instead, I find these tiny little stinkhorns crawling with like flies and slugs because they love the weird smell. And that's the whole purpose of this mushroom is it's spreading its spores by attracting animals to crawl all over it and then move it around. And then you have the yellow staining agarics I mentioned earlier. So these smell like phenol. If you were born before 1991, phenol you will remember as the smell of Band-Aids because they used to put phenol in Band-Aids, but after 91, they stopped doing that. So for younger folks, it would be the smell of library paste or disinfectant or even like a really peaty scotch is the smell of phenol. Basically, if your agarics turn yellow and smell bad while you're cooking them, don't eat those because you will lose your lunch. Okay, so in the book, I come from a wine background, and I had in grad school made a bunch of wine aroma wheels to help provide language for describing wines and training people on how you have the vocabulary to describe wines. When it comes to mushrooms, though, I think a lot of people will smell a mushroom and go, yeah, it smells like a mushroom. Be like, but does this mushroom smell like that mushroom? Like, well, they both smell like mushrooms, but they smell different. So how do you describe those if you don't have a vocabulary? And that was the whole idea of this mushroom aroma wheel was to create a tool using the same logic that I use in wine to have sort of broad categories. Like, well, if it's stinky, is it like musky, fecal, rotting meat stinky, or is it like pungent, smoky ashtray stinky? Like, what kind of stinky are we talking about? So I put all of these terms in here. I talked to a bunch of different mycologists, kind of created a big word cloud, and then figured out like what can I put in here to help people describe the smell of mushrooms. And then around the outside, I also have examples of some of the different molecules, particularly like sotalol and candy caps, as well as benzaldehyde that's in the almond agarics. So kind of a cool little tool for you guys to use. And then the book finishes off with a section called Not Fungi, but Still Fascinating. So these are microheterotrophic plants. They are things that do not do photosynthesis. They've completely lost chlorophyll. They have no green leaves, and they're fully dependent on their association with the mycorrhizal network. So they're kind of like little rhizomes, like ginger-like things in the ground. And they've been thought of as being like parasites on trees and mycorrhizal networks until fairly recently when there's been some work that comes out that says they might be a little bit more like little almost like batteries in the soil selecting for certain types of fungi and influencing like the microbiome of the soil and helping to kind of like be part of the mycelial network by stalking different types of fungi. And there's different associations that these microheterotrophs have. One that is particularly good to know about is the sugar sticks or allotropa. They're associated with matsutake in particular. So if you see allotropa earlier in the year, you can go back later in the year and probably find matsutake in that same habitat. An interesting thing is that all orchids in nature are microheterotrophic at the start of their life cycle. So an orchid seed is the most tiny little speck of stuff, and there's no germ inside of the orchid seed. There's no actual nutrients stored inside of an orchid seed. Instead, when it goes to germinate, it essentially puts out a call to the world around it and calls for orchid mycorrhizal fungi, which is a whole specialized set of fungi that coordinate with orchids. And the orchid kind of hijacks the nutrients from the orchid mycorrhizal fungi to germinate and grow a new plant. And some orchids, like these coral rhiza or coral roots, will remain microheterotrophic for their whole life cycle. And then other orchids, after they've germinated, will then grow green leaves and start doing photosynthesis. But at least initially in nature, all orchids are microheterotrophic, which I think is really neat. And then we have acellular slime molds. So these are not fungi. They are more like protists or amoeba. They kind of crawl around and absorb things. They phagocytose and digest them internally, but you'll see them while you're out hunting mushrooms. And so they start out with this amorphous plasmodium, literally like the blob form that's kind of crawling over stuff, consuming it. And then as they mature, they form these more distinctive sporangia or fruiting bodies, which to me look sort of the coolest in this like semi-immature state. They still have colors, but eventually they all like turn kind of brown and just spore out and will last for, several months as their spores disperse around. But in this state, they look really wild and really cool. So what's awesome about slime molds is if you time lapse them, you can literally see them moving and breathing. This is a phenomenon known as cytoplasmic streaming, as the intercellular components are kind of going back and forth. So every one of these like little clips is at least like 60 to like 120 minutes long. So this short video was like over like 12 hours of my life spent just waiting for these things to kind of sit there and like pulse on the forest floor. Uh, so I think slime molds are really neat and really interesting. So that's why I included them in the book. Um, but thank you guys so much for listening. If you'd like to learn more from me, you can check me out on all the different social medias. Even for people who do not have social media, you probably still use YouTube. So I am on YouTube. You can follow my content there. If you want to listen to more of the stuff I've said about mushrooms, you like hear me talk about science, I have a podcast that's on Spotify and YouTube. And just recently, I started reading my book as an audiobook, as in sort of podcast format, and that's just called Passport to Kingdom of Fungi. You can look that up on Spotify, Apple, Patreon, and YouTube. If you'd like to get some original mush merch or cool t-shirts, I've got that stuff on my website, fastedbyfungi.com. If you want to order cultivation supplies or foraging stuff, northspore.com is a pretty good company. I've got a relationship with them. You can use my discount code, which is fascinated by fungi. And if you have a chance, please pick up a copy of my book, Passport to Kingdom Fungi. You can get it online. I recommend you get it from a small local bookstore, but order it wherever you want. And there's also a companion field guide called Dr. Fungi's Mushroom Journal and Foraging Guide. And this one's a little bit easier to take into the field because it's a little bit lighter, but it has all the basic information on the mushroom, a QR code, and then space to kind of journal and write about your mushrooms. So anyhow, thank you guys so much for listening, and I'm happy to stick around for questions.
Wow. Thank you so much. Holy cow. I want to open it up here. Does anyone have any questions? Gosh, you covered so much. Actually, Eric is talking about doing the audiobook podcast and really highly recommends it.
Thank you.
Yeah.
Yeah, my publisher wouldn't let me do an audio version. They said there's too many photos, there's too many illustrations. I'm like, well, that's a good thing, right? And they're like, well, there's no way you can possibly read it out loud. And I was like, hold my beer. I'm going to read it anyway. So, all right. I see Michelle Shaw's got a question. You want to go ahead, Michelle?
Hi. Yeah, thank you so much for your talk. I am wondering, where do you recommend getting more information about using mushrooms for bioremediation?
See, that's a tricky one because it's a really fun idea, but part of the problem with what's called microremediation is it's not super well defined as a field, and we don't totally know how it works yet. And so that's part of what I was saying. You got to go out to an area with contamination, sample the organisms there, and then grow them, take them back to a lab, do some analysis, understand the chemistry that's happening, and then see if you can select and optimize for something that might be worth reintroducing. A lot of the microremediation is just happening already. right? We're seeing it potentially in some bad ways. Like there's somewhat like 400 to 500 species of fungi that have now been shown to be able to digest plastics, but they're not turning plastics back into like bioavailable soil. They're just turning them into microplastics. So what I said in the book is a lot of these plastic degrading fungi may actually make the problem of microplastics worse before they make it better. There is examples of them using what are called physolithis or dye balls to inoculate trees and then planting them in areas like a strip mine site where people have like pulled heavy metals out of the ground. The ground is completely devoid of life and it's been compacted by, endless amounts of giant trucks and bulldozers and stuff like that. And you can see that if you plant trees without physolithis, they just fizzle out and die. But if you put them there with a physolithis mycelium, that has really strong mycelium that's able to infiltrate into soil. It has high heavy metal tolerance, and the physolithis fruiting bodies themselves can provide about 80 pounds per square inch of force. So they can, I've seen them push up like concrete and asphalt. So they can push up really heavy compacted soils. And so we're seeing that like planting certain types of trees with certain types of mushrooms can help to sort of like be the first step in that rehabilitation of a habitat. But it's not just as simple as like putting something good on a bad thing and getting a good thing back. You got to kind of understand the biology of the system. Hopefully that helps. I think Dave, yeah, sure. Thanks, Michelle. Dave's got a question.
Yeah, I'm wondering if you know if there's a difference nutritionally between a mushroom that you might grow from a mushroom block, like in your basement in a hot box or something, as
Mhm.
opposed to that ones you would harvest.
So in general, I'm going to say that they're pretty similar. Again, to me, the most important parts in a mushroom are like the polysaccharides, is the fiber, and that's going to be pretty similar. When it comes to some of the different organic acids and maybe a couple of the different compounds, that can be a function of like the type of wood that they're growing on. So if you're getting oyster mushrooms that are grown on straw versus something that's grown on like hardwood, you might have a slightly different composition of some of the high order compounds. Which when it comes to something like lion's mane or maybe reishi, you might care more about. But if it comes to just eating mushrooms, I think cultivated mushrooms are going to be very similar nutritionally to what you would get from a wild specimen.
Okay, thank you.
Right. I would note that Christine Solomon, who's one of our members and works at the University of Minnesota, was doing some studies on lion's mane grown in the wild and lion's mane cultivated. And like you said, testing some of those finer compounds to see if they were different. And a lot of them
Mhm.
were the same, but there were some that were different. So yeah, Christine's work would be great to look up. Who else? I think we've got who else is raising their hand? Apologize. Feel free to unmute and ask your question.
Yeah, you can always email me later too at fascinatedbyfungi@gmail.com or just shoot me a message on social media if you have other things that are kicking around in your brain.
Perfect.
Uh,
Dorita?
Jarita, you got to unmute Jarita if you want to ask a question.
Unmute.
Oh, there you
Yeah,
are.
yes, I'm sorry. Very beautiful. Thank you so much. I would
Sure.
like to know more about the medicinal mushrooms. If is any book or any person that I can follow through or where I can learn more
Sure.
about.
Well, I wrote about medicinal mushrooms in my book, so you can read about them there. Uh, I would say that another really good resource to read about for medicinal mushrooms would be Dr. Christopher Hobbs. He wrote a pretty good book about medicinal mushrooms. The problem with medicinal mushrooms is they're often advertised as being like a cure-all, and I will tell you there's no silver bullet for health. I think there is definitely potential for taking medicinal mushrooms, and particularly when you're dealing with something like chemotherapy or your immune system is crashing out, it's a good idea to take quite a bit of medicinal mushrooms, so up to something like 3 to 6 grams a day if you're undergoing chemotherapy. But for a lot of like healthy individuals, you may not need to take supplements, and what might be better is to just eat mushrooms and have mushrooms be a regular part of your diet because you'll get more out of eating them as part of your food than you will out of taking like a little bit of tincture or a really expensive pill every day. But if you want to learn more about medicinal mushrooms, there's quite a bit about them in my book. And if you want to dive even deeper, you can check out the work of Christopher Hobbs.
Thank you so much.
Thanks.
Sure.
Great. Any other questions while we have Dr. Walker? Actually, Peter was asking if he could have a copy or an image of your best practices
Well,
slide.
it's in the book.
There you go. Perfect.
So
That's
I'm gonna throw the link for my guides into the chat one more time, just in case you
perfect.
want to see them. And if anyone's interested in getting a signed copy, I can't do this for you guys in person. I've been in Colorado signing books while I've been here. But if you order from the Napa Book Mine, that's a local business in Napa, I can go down there and personalize copies for you if you want to order one. So, and also I like to support, a small local business rather than like Amazon. But I will say if you're cheap and you just want to get it quick, Amazon is there. So.
Are you at Soma Camp pretty much every year?
Yes, I am a Sonoma Mycology Association board member. I am now the chair of our culinary committee, so I manage all the food at camp and make sure that everyone gets fed well. So I
So,
will be there.
folks, if you want to go to a mushroom event when it's really, really cold here, it's usually MLK weekend in January, and drink a lot of wine, eat a lot of good food, and go mushroom
Yeah,
hunting. I would recommend it's called Soma Camp, and it's the
yeah.
Sonoma County Mycological Association. And yeah, it's a really nice event.
I should also be at the Youp foray this year. I'm not going to BC, but I think I'll be at the Youp one cooking there.
Oh, awesome, awesome. Well, so yeah, that's drivable for us for a change. So we're excited. But perfect.
Rock on. Yeah.
Anything else? Any other questions while we have Gordon? Great, awesome. Well, thank you so much. I would just
Sure.
we were on quite the ride, and it was like just a nice, yeah, it was
In
like
a
I said,
couple years from now, I
good
hope to give talks that are a
information.
little bit more focused on like fermentation and other things. But right now, I got to just share the book. And so thank you guys for giving me an opportunity to share
Oh,
with you.
I want to thank you so much,
For sure.
Gordon. It was fabulous.
Thank you guys for having me.